Right now, in this very moment, a silent battle is being waged. It's not on a distant field, but within the intricate landscape of your own body. This isn't a battle of wills, but of fuel. You are the sole quartermaster of this war, and every decision you make, every forkful of food you take, is directly arming one side or the other. You are either reinforcing the strong walls of your own immune system, or you are, unknowingly, building arsenals for your greatest internal enemy. The most terrifying truth? Most people have absolutely no idea they are the ones tipping the scales. The statistics are jarring, the stories heartbreaking. We are witnessing an unprecedented rise in chronic diseases, with cancer standing as a formidable, relentless specter. It touches lives arbitrarily, leaving confusion and devastation in its wake. But what if there's a pattern within the chaos? What if, beneath the surface of genetic predispositions and environmental factors, there is a fundamental metabolic logic at play? Today, we go beyond the superficial advice. We pull back the curtain on the hidden mechanisms that fuel this disease. We're not just talking about what to avoid. We're talking about understanding the language of your cells, both healthy and malignant. We will dissect the subtle, pervasive habits that act as gasoline on a smoldering fire. And more importantly, I'm going to arm you with a precisely designed, scientifically validated blueprint. This blueprint isn't about just surviving. It's about making your body so inherently hostile to cancer cells that they are starved, suffocated, and neutralized before they can ever gather the momentum to threaten your life. My name is Dr. Liam Cooper. For over 2 decades, I have dedicated my career to investigating this complex relationship. I've worked in the lab analyzing cellular metabolism, at the bedside with patients grappling with diagnoses, and alongside leading researchers trying to decode the fundamental biology of how cancer behaves. What I have learned in my 20-plus years of deep study is something that genuinely shocked me, and it continues to stun my patients when they hear it. Cancer cells are not an anomaly. They are not some external virus that happened to get inside. They are your own cells. They are cells that share your DNA. But they have broken the covenant. They have gone rogue. And here is the truly unnerving part about rogue cells. They are incredibly intelligent. They are strategic. And above all else, they are voraciously, relentlessly hungry. The fundamental question we must confront today is this: are you, through silence, ignorance, or seemingly harmless choices, accidentally providing them with the very fuel they need to thrive? Every single living cell in your body requires energy to sustain life. Your normal, healthy cells are highly efficient, masterfully engineered engines. They take the oxygen you breathe and the nutrients you consume, channeling them through a sophisticated metabolic pathway known as oxidative phosphorylation. This process takes place within your mitochondria, the powerhouses of your cells, and it generates a massive amount of energy cleanly, safely, and with very little metabolic waste. Cancer cells, however, operate on a completely different broken engine. They rely on a metabolic loophole discovered nearly a century ago by Nobel laureate Otto Warburg, a phenomenon we call the Warburg effect. Warburg discovered that even when plenty of oxygen is available, cancer cells abandon the clean, efficient mitochondrial highway. Instead, they pivot to a primitive, highly inefficient process called accelerated glycolysis. They ferment glucose to create energy. Because this fermentation process is so inefficient, a cancer cell requires an astronomical amount of raw fuel to survive and multiply. To compensate, they heavily overexpress insulin and glucose receptors on their cellular walls. They essentially grow hundreds of extra microscopic claws designed to grab every molecule of sugar passing by in your bloodstream. As a result, cancer cells consume glucose at a rate up to 200 times faster than your healthy cells. This is not just a minor biological quirk. It is a profound metabolic advantage that these rogue cells exploit every single time your blood sugar spikes. Every time you consume rapidly absorbing carbohydrates or hidden sugars, your blood glucose surges, and these hungry cells pull that fuel out of circulation before your healthy tissues can even get a turn. But looking strictly at glucose only scratches the surface of modern oncology research. There is a secondary fuel source that is just as critical to a cancer cell's survival, and it is something most people have never heard of: glutamine. Glutamine is an amino acid, a building block of protein. While sugar provides the raw energy for cancer cells to run, glutamine provides the structural building blocks, the nitrogen and carbon they desperately need to replicate their DNA and clone themselves into a tumor. This dual fuel dependency is known in advanced metabolic therapy as sugar and spice. Cancer cells use glucose to power the factory, and they use glutamine to manufacture more cancer cells. When you combine a diet high in refined sugars with a constant overabundance of specific, highly bioavailable proteins, you are accidentally providing these rogue cells with the perfect dual-fuel chemical cocktail. It allows them to grow, divide, and evade your immune system at an exponential rate. Understanding this dual-fuel dependency changes everything. It means we cannot just look at sugar in isolation. We have to look at the entire metabolic environment of the body. We have to look at how our daily choices create a flood of glucose and a surplus of building blocks that allow these rogue cells to build their empires. The power to change this environment doesn't require a miracle. It requires a systematic understanding of what is entering your bloodstream 3 times a day. To truly understand how this metabolic fire spreads, we have to look closely at the chemical messenger that orchestrates the entire process: insulin. Every single time you consume carbohydrates, your pancreas secretes insulin to shepherd that glucose out of your bloodstream and into your cells. But when you overwhelm your system with a continuous flood of refined starches, ultra-processed foods, and hidden sugars, your cells eventually become numb to this signal. This is the hallmark of insulin resistance. To compensate, your pancreas is forced to work overtime, pumping out higher and higher volumes of insulin just to keep your blood sugar from reaching toxic levels. This chronic hyperinsulinemia, or constantly elevated blood insulin, is a massive catalyst for cellular disaster. Insulin is not merely a blood sugar manager. It is one of the most potent mitogenic, or growth-promoting hormones in the entire human body. It acts as a master biological key. When insulin levels are chronically high, it binds to insulin receptors on precancerous and cancerous cells, sending a relentless, high-voltage signal to the cell's nucleus that commands it to divide, multiply, and ignore the body's natural orders to self-destruct. Furthermore, this high insulin environment triggers a dangerous domino effect in the liver, stimulating the overproduction of another volatile hormone, insulin-like growth factor 1, or IGF-1. In a growing child, IGF-1 is essential for building bones and tissues. But in a fully developed adult, high levels of circulating IGF-1 act like premium aviation fuel poured directly onto a spark. IGF-1 activates a highly specific intracellular pathway known as the mTOR pathway. Think of mTOR as the master nutrient sensor and growth switch inside your cells. When mTOR is constantly turned on by a surplus of insulin, IGF-1, and an overabundance of certain animal-derived amino acids, it completely shuts down your body's cellular quality control. Your body stops repairing damaged DNA and instead focuses all its energy on raw, uninhibited cellular manufacturing. This isn't a theoretical concern. Modern epidemiological data shows a devastatingly clear link. When a person develops type 2 diabetes, the ultimate manifestation of insulin resistance, their risk of developing pancreatic, colorectal, endometrial, and postmenopausal breast cancer skyrockets. We are looking at a state of hypergrowth where the natural brakes of the human body have been completely severed by a broken metabolism. To make matters worse, cancer cells possess a remarkable ability to adapt when glucose drops slightly by aggressively scavenging for a third fuel source, circulating free fatty acids. In a heavily compromised, metabolically unhealthy body characterized by visceral fat accumulation, the dangerous fat wrapped around your internal organs, the body is constantly leaking these free fatty acids into the blood. Rogue cells readily absorb these lipids, utilizing a process called fatty acid beta oxidation to fulfill their massive energy quotas. They are metabolic shapeshifters. If you feed them an abundance of sugar, they feast. If you pair that sugar with a state of chronic insulin resistance and elevated fats, you provide them with multiple backup generators, ensuring their survival even under stress. Fueling cancer is not just about what you feed the cell directly. It is also about the climate you create inside your body. The second massive catalyst for malignant growth is chronic, low-grade systemic inflammation. To understand this, Imagine your immune system as a highly disciplined internal defense force. When you get a cut or an infection, your body releases a localized burst of acute inflammation, temporary heat, swelling, and chemical signals to destroy invaders and repair the tissue. Once the job is done, the defense force stands down. But what happens when the fire trucks never go home? What happens when a continuous stream of chemical triggers keeps your immune system on high alert for months or years at a time? This is chronic inflammation, a state where your body is silently burning from the inside out. The primary architect of this internal fire is our modern industrialized diet, specifically the overconsumption of ultra-processed foods and highly refined industrial seed oils like soybean, corn, and cottonseed oils. These oils are packed with omega-6 polyunsaturated fatty acids, specifically linoleic acid. When consumed in the massive quantities found in packaged foods, restaurant deep fryers, and commercial snacks, they overwhelm your body's anti-inflammatory omega-3 pathways. This imbalance forces your tissues to produce a relentless cascade of pro-inflammatory signaling proteins called cytokines, such as tumor necrosis factor alpha, TNF-alpha, and interleukin-6, IL-6. In a highly advanced oncology review, researchers noted that these inflammatory cytokines do something deeply sinister. They actively mutate your healthy DNA. They create a molecular microenvironment that acts like a warm, protective incubator for rogue cells. Under the constant assault of chronic inflammation, your body's natural DNA repair mechanisms, the cellular editors that fix genetic typos, become completely exhausted and shut down. Even worse, this inflammatory environment alters the behavior of your immune cells. Macrophages, which are supposed to hunt down and devour abnormal cells, become hijacked by the tumor's chemical signals. They transform into what we call tumor-associated macrophages, or TAMs. Instead of killing the cancer, these corrupted immune cells begin to build physical scaffolding for the tumor, shielding it from your body's natural killer cells and secreting growth factors that help the tumor expand. This brings us to a critical milestone in cancer progression: angiogenesis. A microscopic cluster of rogue cells cannot grow larger than the size of a grain of sand without a dedicated blood supply. It runs out of oxygen and nutrients. To overcome this, the tumor uses the surrounding inflammatory environment to send out chemical distress signals, primarily a protein called vascular endothelial growth factor, or VEGF. This protein commands your existing blood vessels to sprout entirely new, abnormal branches directly into the tumor. Once this independent pipeline is established, the cancer is no longer a localized hitchhiker. It is a fully integrated metabolic parasite linked directly to your main blood supply, ready to drain your glucose, glutamine, and fatty acids at an accelerated rate. While the internal landscape of inflammation and hormones dictates much of this battle, we must look even deeper into the literal core of your digestion, the gut microbiome. Your gastrointestinal tract is home to an ecosystem of trillions of microorganisms that dictate up to 70% of your entire immune system's daily function. In a healthy state, these beneficial microbes act as strict drill sergeants for your immune surveillance. They train your natural killer cells and T cells to patrol your tissues, spot mutated rogue cells, and eliminate them long before they can aggregate into a tumor. However, our modern lifestyle creates a state of severe microbial imbalance known as dysbiosis. When your diet is stripped of complex plant fibers and heavily loaded with artificial additives, preservatives, emulsifiers, and excess alcohol, the beneficial, protective bacteria starve to death. In their place, opportunistic, pro-inflammatory species proliferate. These pathogenic bacteria degrade the mucosal lining of your intestines, creating microscopic gaps in the cellular wall, a condition known clinically as increased intestinal permeability or leaky gut. When this barrier breaches, toxic bacterial cell wall fragments called lipopolysaccharides, or LPS, slip directly into your portal vein and flood your systemic circulation. Your immune system recognizes LPS as an existential threat, triggering a massive body-wide inflammatory response. This constant low-grade endotoxemia acts as a continuous cloaking device for cancer cells. because your immune defense force is completely distracted fighting the ghost fire of circulating bacterial toxins. Its ability to perform high-level immune surveillance drops dramatically. Rogue cells are left unpoliced, free to establish themselves and divide. Furthermore, an unhealthy microbiome directly alters your body's hormone clearance through an architectural network of specific bacterial genes known as the estrobolome. The estrobolome produces an enzyme called beta-glucuronidase. When your gut is compromised by dysbiosis, these bacteria produce an excess of this enzyme, which uncouples bound estrogen that your liver was trying to excrete. This free, active estrogen is reabsorbed straight back into your bloodstream. For hormone-sensitive tissues like breast, ovarian, and endometrial tissues, this creates a state of constant estrogen dominance. Estrogen, much like insulin, acts as a profound cellular proliferator when left unchecked. It drives the rapid replication of cells in breast ducts and uterine linings. If a rogue cell with an estrogen receptor is present in that tissue, this recycled hormone acts as a direct growth signal. The data coming out of advanced microbiome sequencing projects is undeniable. A damaged gut doesn't just cause digestive discomfort. It actively dismantles your body's primary shield against metabolic and hormonal oncogenesis. Now that we have comprehensively exposed the exact cellular mechanics, hormonal cascades, and microbial pathways that feed and shield these rogue cells, we must shift our focus to the solution. We must look at how to systematically reverse this microenvironment. We do this by implementing precise, scientifically validated strategies designed to shift your body from a state of hypergrowth to a state of deep cellular repair and metabolic hostility toward disease. The cornerstone of this defense strategy is the deliberate, strategic activation of a profound cellular process called autophagy. Rooted in the Greek words meaning self-eating, autophagy is your body's internal recycling mechanism and quality control system. When your cells are constantly bombarded with nutrients, specifically glucose and insulin, the master growth switch, mTOR, remains turned on, completely blocking autophagy. However, when you intentionally create a structured window of nutrient scarcity through intermittent fasting, the drop in insulin triggers a dramatic cellular pivot. Deprived of an external influx of easy fuel, your healthy cells turn inward. They begin to systematically dismantle, digest, and clear out damaged mitochondria, misfolded proteins, and precancerous cellular debris. Healthy cells are highly resilient. They easily survive this temporary pause by switching to clean-burning ketones for energy. Rogue cells, however, are metabolically rigid. Because of the Warburg effect we discussed earlier, they cannot easily adapt to burning ketones. They depend on high volumes of glucose. When you clear your bloodstream of excess glucose and suppress insulin for 14 to 16 hours, you are quite literally starving the enemy while simultaneously forcing your healthy cells to clean house. This elegant biological reality is why fasting-induced autophagy has become one of the most intensely studied areas in modern preventative oncology. Beyond nutrient timing, we must look at the specific biochemical compounds found in nature that can actively jam the signals cancer cells use to survive. We don't just want to restrict their fuel, we want to actively disrupt their infrastructure. Consider the compound sulforaphane, found in exceptionally high concentrations within cruciferous vegetables like broccoli sprouts, kale, and Brussels sprouts. Sulforaphane has been shown in molecular studies to directly target cancer stem cells, the highly resilient core cells responsible for tumor recurrence. It works by down-regulating the intracellular pathways that allow these cells to hide from your immune system, while simultaneously upregulating your body's master antioxidant pathway, known as NRF2. Similarly, we can look at the active compound in turmeric, known as curcumin. At a cellular level, curcumin acts as a broad-spectrum signaling disruptor. It directly interferes with nuclear factor kappa B, NFKB, which is the master genetic switch that turns on chronic inflammation inside your cells. By blocking NFKB, curcumin effectively dampens the production of those pro-inflammatory cytokines that mutate DNA and create a protective cocoon around rogue cells. Furthermore, we must intentionally combat the process of angiogenesis, the tumor's ability to grow its own blood supply lines. Epigallocatechin galate, or EGCG, a powerful catechin found abundantly in green tea, has been shown in peer-reviewed trials to directly inhibit the signaling molecules, like VEGF, that tumors secrete to recruit nearby blood vessels. By introducing these targeted botanical compounds alongside a low glycemic, fiber-rich dietary matrix, you are effectively executing a multi-pronged assault. You drop the blood sugar to starve their engines, block inflammation to strip away their shield, and inhibit angiogenesis to cut off their supply lines completely. To fully solidify this internal fortress, we must look beyond nutrition and address the systemic lifestyle pillars that dictate your daily cellular environment: physical movement, sleep architecture, and stress physiology. Each of these pillars exerts a direct, measurable influence on your hormonal baseline and immune surveillance capability. Let us first examine the mechanical and biochemical impact of targeted physical activity. When you engage in regular exercise, particularly skeletal muscle contraction through resistance training or zone 2 cardiovascular movement, you are doing far more than burning calories. Your muscles act as a massive metabolic sink for circulating glucose. During and after physical exertion, your muscle cells deploy glucose transporter proteins directly to their walls, absorbing sugar out of your bloodstream without needing a massive surge of insulin. This instantly lowers your circulating insulin baseline, shutting down the primary mitogenic growth signal we established earlier. Furthermore, intensive physical movement acts as a direct stimulant for your immune system's frontline soldiers, natural killer, NK cells, and cytotoxic T lymphocytes. The physical shearing force of increased blood flow mechanically flushes these specialized immune cells out of storage sites like the spleen and pumps them directly into your peripheral circulation. This drastically heightens immune surveillance, allowing your body's natural hunter-killer cells to aggressively scan your tissues, identify rogue cells, and eliminate them via targeted apoptosis before they can ever aggregate into an invisible tumor mass. The second overlooked pillar of systemic defense is the architecture of deep sleep. Sleep is not a passive luxury. It is an active, highly regulated neurological and endocrinological repair phase. During deep, slow-wave sleep, your brain's pineal gland secretes peak levels of the hormone melatonin. While widely recognized as a sleep regulator, modern cellular oncology recognizes melatonin as a premier, highly potent endogenous antioxidant and oncostatic agent. Melatonin possesses the unique structural capability to easily cross cellular membranes and enter the mitochondria, where it directly neutralizes the free radicals that cause catastrophic DNA strand breaks and subsequent oncogenic mutations. When your circadian rhythm is chronically disrupted, whether through artificial blue light exposure late at night, poor sleep hygiene, or shift work, your nocturnal melatonin production collapses. This drop strips your cells of their nighttime molecular repair cycle. In fact, the epidemiological data connecting chronic circadian disruption to heightened cancer risk is so robust that international health authorities have long classified night shift work as a probable carcinogen. Prioritizing 7 to 9 hours of quality sleep in a completely dark, cool environment is a foundational, non-negotiable step in maintaining cellular integrity. Finally, we must confront the modern epidemic of chronic psychological stress and its hormonal proxy, cortisol. When you exist in a perpetual state of psychological or emotional survival, your adrenal glands continuously secrete cortisol and catecholamines like adrenaline. Chronically elevated cortisol acts as a profound immunosuppressant, directly shrinking your thymus gland and dampening the production and efficacy of your T cells. Simultaneously, adrenaline binds to adrenergic receptors on rogue cells, activating intracellular signaling cascades that promote cellular survival and tissue invasion. Chronic stress, quite literally, creates a state of systemic vulnerability, paralyzing your internal fire department while creating an optimal, unpoliced environment for abnormal growth. Managing this nervous system stress through deliberate breathwork, mindfulness, and structured rest is not an abstract concept. It is a direct physiological mechanism to downregulate the chemical messengers that allow rogue cells to multiply in the shadows. We must look at the hard truth of modern oncology. Cancer is undeniably complex, but we are far from helpless in the face of it. For over 2 decades, as Dr. Liam Cooper, I have analyzed the deep metabolic pathways that control cellular life, death, and mutation. What the clinical data clearly reveals is that up to 40% of all cancers are completely preventable through deliberate, targeted lifestyle choices. Nearly half of this battle rests squarely within your daily control. Every single meal you consume, every hour of deep sleep you prioritize, every walk you take, and every stress signal you downregulate is a direct vote for your long-term cellular destiny. You are either actively feeding the broken metabolic engine of a rogue cell, or you are reinforcing the sophisticated, highly intelligent defense network of your own immune system. Do not wait for a clinical diagnosis, an abnormal scan, or an unexpected health crisis to start paying attention to the internal environment you are building. The optimal time to construct a highly resilient, metabolically hostile body is right now, today, before the alarm bells ever ring. Start by ruthlessly eliminating added sugars, optimizing your gut with fiber and fermented foods, moving your body daily, and protecting your circadian sleep cycles. These choices are straightforward, but their biological impact on your DNA repair mechanisms is absolute and measurable. Please be advised that the insights shared in this video are for educational and informational purposes only. This information does not constitute medical advice, diagnosis, or treatment, and should never replace consultation with your personal healthcare team or oncologist. If this deep scientific breakdown brought you true value today, please take 2 seconds to hit the like button, leave a comment with your thoughts, and subscribe to the channel with notifications turned on. Take care of your health, protect your cellular biology, and I will see you in the very next video.